Thank you, Marty. And you hit on a number of really important things. One of them being the reproducibility crisis. So I'd like to talk today not only about Thrive, but about that incredibly important problem, which many on this call may not know about, but should be aware of because it affects drug development and us getting medicines to help us. And it's the reproducibility crisis, the ability to repeat research. It's astonishing how little research is repeatable because so many of the processes are manual. And we started Thrive in the Boston area because we found this ignored but critically important area, which is manual cell culture without data, without automation, without documentation. And so much is built on it. And it's really holding back and hurting the odds of success for drug discovery. Thank you, Marty, for having me. This is my third one. I look forward to seeing you in Austin, in New York as well. So my own background, I've started 11 companies in addition to having run several public companies, 3 of them in software and the remaining 8 in life sciences. And compared to the people that you mentioned, I'm humbled. But I have had 4 multi-billion-dollar exits and changed the course of cervical cancer. We changed how cytology labs read pap smears and now have 95% market share in the US. I started a company called Exact Sciences, public company, which has a replacement for the colonoscopy. And chances are when you, if you got the vaccine, it was my syringe, my safety syringe. I sold that company to Becton Dickinson, and it's now the world's most widely used syringe. So I'd like to talk today about the products that we make and the problems we're solving. And just to put it in kind of brief terms, we make products for researchers, and particularly for drug discovery, to more effectively study living cells through imaging, analytics, and automation. It is an experienced team. I'll talk about that in a bit, but my co-founder is responsible for the sequencing instruments that Illumina has, which are the most widely used and has brought data to genomics. And our chairman ran the largest laboratory products group in the world at Thermo. We have incredible collaborations, not only with a large pharmaceutical company in Japan, but with truly the best leading research centers. Harvard Stem Cell Institute, Massachusetts General Hospital, the Broad, University of Texas Medical Branch, Galveston, leader in infectious diseases. And because these instruments that we make haven't been made before, and there's been such limited innovation in the field, we have 78 patent applications, of which 25 have issued. So we have, we've sold early access instruments. We have about $875,000 in sales to date, and we're launching our products now and expect $6 million in revenue this year, growing to over 150 million in 5 years. And I have 3 other companies that have grown faster than that, actually. We've raised $29 million to date from mostly family offices. Our largest investor is the Estee Lauder family office. They invested in my second company, which was a multi-billion-dollar exit. And then they invested in Cytic, which we sold for $6 billion, and which changed how cytology labs work. Also the Milstein family, real estate family in New York, Parker Brothers, which the family sold the business to General Mills, famous for making Monopoly. And they actually invest a lot in life sciences, almost exclusively. So I'm spending Monopoly money. Something I dreamed about. I used to dream as a kid that was real money. And we also have 5 angel groups, including the largest one in the world, Life Science Angels, and industry veterans, such as former CEOs of companies like Novartis. And we're currently raising a $5 million convertible note that will convert into our Series B, which we expect to have mid-year. It will convert at a 20% discount. And it's basically to expand our sales and marketing and to acquire a small profitable company that is also in this same space. So cell culture is not something that everybody knows about, but it is key to biomedical research. It is a huge market. It's $29 billion per year. It is conducted extraordinarily manually and without data, just like it was 65 years ago. And the way it works is that researchers spend a couple of hours a day in the lab. They take the plates out of the incubator, bring them over to the microscope, spend on average 2 hours looking at millions of cells, hundreds of thousands of stem cell colonies, and then they put the plate back into the incubator, and guess what? All the data is lost. We know so little about the cells that are the basis for development of drugs. Uber knows more about us than we know about the cells. They collect far more data. And it's not just what is happening today in the lab. As a result of this being a manual process, the NIH and FDA did a study and found that 14% to 33% of cell lines are mislabeled or contaminated. This is extraordinary, and it is one of the many causes for failure of drugs. What this means is a researcher is studying and screening a drug on the wrong cell line, on pancreatic cells instead of on liver cells. And when cells get contaminated, there's an exchange of DNA, so they behave differently. And then, Marty, we take these cells that are of poor quality, highly variable, non-comparable, and then we spend billions of dollars on other instruments and running experiments on these cells, which is why this area is so important. So Nature magazine recently did a survey. 1,500 scientists lift the lid on reproducibility, the crisis rocking research. It all started with Amgen, and it's, I read an article in 2013 about this bombshell study. Amgen tried to replicate 53 landmark studies, which were important to their own research, and they could not replicate 90% of them. And yet they had paid millions to in-license and to perform many of these studies. They had 100 scientists, up to 100 scientists, in over 10 years, and they tried to reproduce the results, and they couldn't. And other studies have found that 50% of the causes of not being able to replicate these, mostly cancer studies, in the case of Amgen, was because of cell culture. And others have shown the same. Study after study, Bayer did one. Nobody has shown that more than 50% of the research that's been done in life sciences is reproducible. And if it's not reproducible, it's not science. And unfortunately, not only does this slow cures, papers cite previous papers and then it gets recited. So these non-reproducible studies cumulatively increase our ignorance. And unfortunately, patients are exposed to clinical trials in drugs that are based on not reproducible research, several 100,000. So our solution is instruments and software that provide advanced imaging, analytics, automation, I should add documentation, for reproducible cell culture, stem cell culture, and tissue culture. Massachusetts General Hospital said about our instrument, this is what cell culture should look like. With cell assist barcoding, we no longer mix up plates, forget which reagents we used, guess at who did what, and lose valuable time and money. And that's just one part of what we do, which is our documentation system. So we have a family of products. We have two major products on the market, even though we're at an early stage. And what's important about all these products is, you referred to it, Marty, as binary. They're not binary. So we do not require FDA approval or clearance. This is the first company in decades where I have not had the constraints of having to worry about FDA approval at every turn. We're selling into the research labs, and even though we're getting quickly pulled into bioprocessing and the manufacturing of cell therapies, even there we are not directly related. We merely need to show that we meet certain standards of how we handle data. So on the bottom left is our first product. There's actually one behind me. It's a benchtop unit that takes hundreds of thousands of images of cell culture plates very rapidly. and processes them, creates a database, analyzes them. Just to the right of that, the Cell Assist 50, which we're going to launch hopefully at a conference in February, takes the imaging from our benchtop and combines it with some robotics with 50 plate incubators. So it can around the clock image plates and remotely. But most of our focus right now is on software modules. Of our 10 R&D people, 9 are actually software. And we're really helping our, I'll tell you more about rapid viral plaque assays. We're really helping our customers have breakthrough insights. And our next major instrument will be full walk-away automation. So probably the most important of the secret sauce, although there are many in what we do, is our imaging. We're the only instrument that can actually find and track a single cell or colony over time. That means this is a desire that every researcher has had, to be able to follow a cell over time, see how it reacts to drugs, has never been able to be done before. And there are many workflows such as CRISPR, where you only have a single cell that you've engineered. And right now, people can't even see them for 12 to 14 days. They have to wait till they've doubled enough that they can even find them under the microscope. We're really a 4th generation microscope. And unlike other instruments, we capture between 5 and 60 focal planes, 2.5 microns to 50 microns apart. This allows us to image organoids, to get the sharpest images of any instrument, and to see processes happening in the cells that nobody else is able to capture. And we image all the cells in all the wells and all the plates in the lab. And you really do want to see all the cells if you're putting them back into a research subject, and particularly a human. So we basically, in one scan, which takes tens of minutes, we capture up to 30,000 images. That's 90 gigabytes in 20 minutes. Our labs, our sites are producing 2 to 3 terabytes per week. Live cell biology has never had this kind of data. And it's really bringing genomics quantities of data to live cell biology. One of our customers said, who had some breakthroughs studying COVID-19 with our instrument, Thrive will change the world. And I said to him, can I quote you on it? I thought he was joking. He said, you can. And it's because Thrive allows us to see and understand living dynamic cells and tissues in 3 dimensions over time with remarkable images. We're really closer to the biology. So you can actually follow what happens, and you can see it in almost 3D. And we provide benefits for the lab managers in terms of increasing yields, saving money, reducing errors. And we help researchers for the first time in live cell biology. There are data sets that can be used for machine learning and for data mining. It just hasn't been available before. And most of our sites have told us they've had breakthrough insights. So we also help with the mundane. I mentioned documentation. I mentioned 14% to 30% mislabeling and contamination, and most people can't even measure it. It's just an accident if they see it. But with data, you're able to see and look at outliers. And this is actually from one of the major research institutes that I mentioned. And we noticed that there were some cells that were off the growth curve, and asked them what was going on there. And they looked into it, and in fact, they were mislabeled plates. And they never would have known that if it were not for our instrument. So one of the breakthroughs comes from a person who's chairman of the biochemistry department at University of Texas Medical Branch, who said that Thrive will change the world. And it's because he is able for the first time to study viruses with new information that hasn't been available before. Basically, the standard is you put an overlay of the virus. This was done with COVID-19 and with every virus, an overlay of the virus on the cells in a plate. And currently, without the cell assist, you have to wait for 7 days and then you have to wash them. So you have to touch it again and stain them. And so you have no data before the formation of these plaques or holes in the cells, and you have no data after because you've stained them, you've killed them. And the reason that he said this is such a game changer is for the first time, you can actually see the progression of these plaques. And he has found that there's important data in there. In fact, he discovered he had a variant when he was screening drugs for COVID-19 that was confounding his data. We are the best, we have the best instrument for imaging tissues because of all these focal planes and the quality of our images. And tissue culture is the fastest growing area in cell culture. So currently we have these plates of cells sitting on the bottom of the plate, and then we have animal data. And there's a big gulf in between for testing drugs. And tissues and organoids are now an area where we can get more information about how the drug affects something that's closer to the tissue. This is from Harvard Stem Cell Institute, and I mentioned we're able to find one cell sitting in this plate and then track it over time so that you can really study the processes. This is a tumor model, an organoid from Massachusetts General Hospital. And they also said for the first time, they could study essentially the shedding of cells from the tumor. So we have the team to do this. I told you a little bit about my own background, starting companies in software and mostly in life sciences. A very close comparable is Cytic, where we automated cytology labs. And we sold Cytic to Hologic for $6 billion. And now 95% of all pap smears are done by our instrument. My co-founder, is responsible for most of the genomics workhorses, which are sequencing instruments. He started off in Lee Hood's lab, and many people don't know that Illumina, who's by far the leader in sequencing instruments, actually didn't develop them themselves. Alan built them at a company that they acquired. In fact, most of the major innovations have come from small companies and then are acquired by larger companies, which will happen to us. Chair of our board was president of Thermo Fisher, the largest laboratory products group in the world. Mike Finney's in San Francisco, started several companies. And our third outside board member, Brock Reeve, is executive director of the Harvard Stem Cell Institute. That's an incredibly important application. It's probably the area where information is needed the most. Brock Reeve, by the way, is Christopher Reeve's brother, who died from a horseback injury, but was actually having really good luck with stem cells before he died. He did not die because of stem cells, and Brock has dedicated his life to studying stem cells at the Harvard Stem Cell Institute. So I mentioned we raised $29 million to date. We've had nice increasing pre and post money valuations. We're currently raising a convertible note that will convert into the Series B mid-year. We have an acquisition opportunity of another company that has 6 million in revenue, is profitable, and is within our space. In fact, they're a vendor, and they're the leading company in environmental controls, specifically for cell culture. We need about $5 million and raise it within a month. So if anybody has any ideas, this is a really great opportunity for us. We have a large amount of interest from our current investors. I think most of it will come from them, but we still need some financing for that, and quickly. It'd be great if we can acquire it, but we have a great business as well on our own. I think we're looking at, you never can tell, but I think we're looking at probably an exit in a couple of years. We fit into so many companies. And life science research tools have extraordinary multiples. It's a small segment, and so it's not nearly as well known as therapeutics and diagnostics and devices. but we sell them the tools that they need. And whether or not a drug ends up being approved by the FDA, they still need tools like what we provide. And we don't have the FDA risk, which lets me sleep a lot better at night and is a huge de-risker. The multiples for companies under 300 million in revenue in life science tools is 10x to 35x, trailing 12-month sales. And M&A is at 10x because a lot of companies that are failing are sold. And 35x is for publicly traded because they went through a filter. But these are really fantastic valuations. And we've been contacted by 6 or 7 companies repeatedly for potential acquisition, but it's really too early. In a couple of years, we'll have a lot of papers that are out there. We'll be at an inflection point for selling at a much higher price. And we're like many public companies, although we are in our own unique field, there are many life science research tools companies that are selling at 30 times multiple. So maybe just in summary for the last minute, and hopefully you're thinking of some questions. So cell culture is key to biomedical research, yet lacks sufficient data and automation. It's one of the few remaining parts of life sciences which has no tools, no data, no databases. And we sell 2 instruments for researchers and drug discovery that automate imaging and analytics for repeatable, comparable results, cost savings, and breakthrough insights. We have 15 collaborations and customers from industry and research. I mentioned due to limited competition, we have 78 patent applications, of which 25 have issued. We project $6 million in revenue this year. We're just now have hired our first 2 salespeople, and we expect to grow to 150 million in 5 years. We've raised $29 million from well-known family offices and angel groups, family offices like Estee Lauder family. And we're targeting a Series B, $15 million Series B financing mid-year. We actually have a term sheet already for that. And as I mentioned, a leading mRNA and infectious disease biologist and a customer stated that Thrive will change the world. And another leading stem cell researcher said, this is what cell culture should look like. So Marty, we're trying to bring analytics and automation and better biology to life sciences. And I appreciate the opportunity to have spoken. Thank you.
Tom, some questions, don't go anywhere. So I think what you're describing in the lab is that it's not really a Six Sigma process.
Oh God. First of all, they wouldn't even know what that means. And we provide the first barcoding system in cell biology so that in a lab you can barcode the plates and the person and the instruments you use. And they think that's astounding technology. So yeah, it's on the opposite end of the spectrum.
It might be a 1 sigma or a 2 sigma.
Yeah, half.
Right. I mean, there's actually a lot of interesting studies for people who have come out of 6 Sigma programs, and you know, I think they say human error is 2 sigma, right? It just is a nature of things. Even for people who are like dedicated, that's you know the amount of errors that you see. So it's really a fascinating area. Can you talk a little bit more about the payor? Like who pays for this? Is it like labs, people receiving government grants, other entrepreneurial ventures, and like what the costs are to the labs to acquire it, and what's the market size, market opportunity?
Sure, great question. So it's a $29 billion per year market. 80% of that market is drug discovery. So it's pharma and biotech. That's our 80% of our market, and 80% of our 12 sales so far, which were early access instruments, were pharma and biotech. We sell to research labs, and we're beginning to sell to cell therapeutic companies. We have none of the issues of reimbursement. Not only, we don't have FDA approval issues, we don't have reimbursement issues. So these are instruments and software that come out of basically drug discovery. which has enormous budgets. It's not a price sensitive market by any means. And we have instruments ranging from 100,000 to 250,000, and our software modules average about $35,000 each, and will be sold on a subscription basis. So we'll have
the software. It's a software as a service product.
Not so much as a service, but a subscription basis. We are looking at a real software as a service model as well for some of it, but it's really their software that they'll use and tailor to the way that they need it.
So the data is not in the cloud; it's in-house.
So we retain rights to that data. And one of the things I did at Cytic, which really is why we got 95% market share among cytology labs, and I expect to do the same here, is we maintained rights to the data and allowed users, as long as they were a user, to look in a limited way at other people's data. Unfortunately, by the way, just in terms of the cloud, the pharmaceutical industry is one of the last to be adopting the cloud. They're very paranoid, maybe for good reasons. And so all of our sites, we sell them lots of add-on memory. I mean, hundreds of terabytes, or they also use their internal servers, but they allow some data outside of the labs, but not all. But a big opportunity for us is this database of cells, which we'll monetize. We need a few years more to have a larger install base before we can monetize that.
Right. Just to do a reference point for that, I'm going to say that somebody recently sold a database of certain ethnicity of oncology patients for billions of dollars. Right. So as you collect this kind of data, it becomes an incredible, framework. I mean, you talk about there's a value of having the right results in the lab, and then there's the year after year consolidation and collection of data, which is just so valuable for research and other purposes.
Absolutely. For the first time, they can go back if they see something. They can actually go back and not only see what they did and know what reagents they used, but they can compare it to, gee, are there other cases where we've seen cells react like this? And pretty soon we'll let them compare across labs.
Yeah, that's really fascinating. So if you also talk a little bit, so one of the problems with, we've done so much healthcare in this group, one of the problems we always run into is people who have an invention that is like a device or technology, and a lot of healthcare is, I'm not sure it's the right word, disparate, or it's not consolidated where there's, few buyers. What's the state of the lab industry? Are there many labs, or are there a few big players? If you could sell to like one big lab player, they could go and buy, thousands of these orders. Can you talk around that?
Sure. So there are a lot of labs, something like 80,000 in the world, and we expect to get something like 15% or so as available market. But you have some divisions. So you have extraordinary amount of research being done by pharmaceutical and biotech companies, and it's reaching really extraordinary rates, and they're quite they're not price sensitive. And our instruments, although they can support several researchers each, once you have it, you have a level of data that you want to be able to compare across labs. And once one lab has it, other labs want it. So in fact, there are multiple sales to a lab, because basically each researcher or a couple of researchers would want it. There are CROs, contract research organizations. We just sold an instrument to the largest CRO in the US, just bought by Thermo. And just one group that has our instrument has 400 plates, cell culture plates, and that's not uncommon. And so what they really need is our 50 plate incubator that sells, combined with their imaging, that sells for 250,000. And all of our instruments have paybacks 18 months to 24 months. It saves so much time from researchers looking. So there we're looking at, hundreds of instruments. And in fact, we're talking with them about a volume purchase order. There are then large institutes, which are multiple buyers. And then there are small principal investigators, academia, where it's one person. And we actually have products that are for small, medium, and large labs. But obviously, those researchers that are in institutes where they buy multiple is a sweet spot in academia and pharmaceutical drug discovery. So we're looking at large numbers of sales to each of the customers, but they all start with one.